Hello and good evening, good afternoon, good morning, wherever you are in the world. I'm Dr. Philip McMillan. I'm a physician in the United Kingdom. I have been focused on COVID since early 2020, especially the autoimmune mechanisms. Part of my journey has been to speak with international experts all the way from then. And this journey hasn't stopped. It's an ongoing journey because we have so much to learn by sharing and working with each other. So the format for today is very simple. We have 3 speakers, we have 2 panelists. Each speaker will speak for about 20 minutes, and then there will be a question and answer from the panelists, and I will look out for any relevant questions in the audience that maybe can be added to augment the discussion. After we've gone through each speaker, then the panelists discussing with them, next speaker, panelist, we will then have a round table discussion. And that's when we will sit down with each other and it will be freely open. And at that time, certainly we should be able to pick up more of your questions or comments. We appreciate very much everyone who is already with us, and we love it when you share with us where you are from in the world, just so that everybody knows that this is a global audience with global reach. So without further ado, I'm going to be adding the speakers and panelists to the stage, and they will then do a quick introduction of themselves before we hear a short word from our sponsor. So without further ado, let's add all of the people here with us today. Thank you all very much for joining us. I will be asking if you could just do a short 30-second introduction of yourself. And we will go through, we'll start with Manan, then Kevin, then Diane, then Philip, and then Shankara. So without further ado, Manan, could you let people know a little bit about yourself?
I'm Abdul Mannan Baig. I've a PhD in biomedical sciences, and I got involved into COVID research in early 2020. Me and my team were first to publish that the loss of smell and taste actually is the earliest sign of NeuroCovid, then adapted by FDA, NHS. They started segregating patients into two groups based on that. I have worked in St. George's Hospital with Dr. Beate Yeager, and now I have got my own scientific lab in Heidelberg. So all research on COVID and NeuroCovid, I'm dedicated to it. Thank you very much for coming and joining us.
Wonderful. Thank you. And Kevin, what about you?
Yep, I'm a PhD in systems neuroscience. I was very early into COVID. I was one of the first to publish that the agent itself was a chimeric agent, synthetic, likely a consequence of biowarfare research. I'll touch on that a little bit in the presentation. And part of their weapon mechanism was to leverage amyloidogenic cascades in the body, and I've spent the last 5 years digging into that and trying to understand and find countermeasures to what they've done.
Wonderful. Thank you, Kevin. And Diane?
Yes, I'm Diane Counce, and I'm a neurologist out of Birmingham, Alabama. And I have been treating COVID initially, and now I am seeing a lot of long COVID and long vax injury patients. And I've been treating them extensively and trying to keep up with all the latest data and coming up with, supplements and other treatments, and especially neuroimaging using NeuroQuant to identify neuroinflammation and target the brain in particular as far as trying to help heal.
Wonderful. Thank you. And Philip?
Yes, I'm a private practice radiologist here in Birmingham, Alabama. And for the beginning of the pandemic, I kind of jumped out of my wheelhouse and started help treating patients and gained experience doing that. But over the last 5 years, I've gained quite a case of COVID and vax-related radiological cases, which I've compiled over the last 5 years, and have kind of noticed a pattern. And in the uptick in what we've seen. I'm currently helping, actually in Dr. Counce's lab, treating some of our neurological vaccine-injured patients.
Excellent, thank you. And Shankara? You're muted, Shankara.
Hi, everyone. I'm Dr. Shankara Chetty. I'm a natural science biologist and general practitioner. I've been a frontline doctor from the start of COVID, trying to understand the pathophysiology of what transpired. I think with the understanding of spike protein being the primary pathogen, that translated into an understanding of vaccine injuries and the pathologic mechanisms underlying it. So I'm on the front line seeing what everyone's researching in a clinical format, trying to make sense of what's going on in this patients. So I'm glad to be part of the panel asking questions today.
Wonderful, thank you. Just as a point, when we are talking, I think, Manan, if maybe your microphone may be a little bit far from you, so it sounds a little bit distant. I don't know if you can adjust that when you're going to do your presentation. But what we'll do is now we're going to hear a quick word from our sponsor. Thank you all very much for the time being. Next, what we will do is bring Manan on to do his presentation in just a few minutes. So thank you all for being with us, and let's hear a quick word from our sponsor.
And so when COVID started on day one, that is January 2020, I think around the 18th, we dropped everything. And ever since that day, we've been working 7 days a week, practically around the clock on finding solutions. Everybody on that slide you see there, to some degree, are working almost either on a weekly or daily basis, or even in direct collaborations, in research collaborations, in publications, in lab research. And so, you know, each one there you can see has significantly contributed either to treatment or to development or understanding the disease and other discoveries. And so it's a great honor for me to be part of that and to be able to mediate between all these groups. I'm kind of like the coordinating a little bit between all these domain experts.
Thank you very much. And we'll go straight into our first presentation. Manan, just let me hear what you sound like.
I've moved close to the screen, so I think I should be fine.
Oh, excellent. Wonderful. So I'll let you get straight into the presentation. You just let me know when you want each slide to move forward.
Sure.
Okay.
Please. You can proceed with the slide number 1, please, Philip. It's at the top, not this one.
Okay. Sorry, 1 second. I'm just going to this one here. Here we go.
So this presentation, okay, would definitely talk about the NeuroCovid and the effects of this virus on the brain, but it actually reminds me of 5 years back, okay, when me and my team, okay, actually first alerted the globe that this virus is causing loss of smell and taste, and it has got proposed neurotropic mechanisms that we gave in this paper, and it has clearly a neurotropic virus. Then started the gaslighting from my peers where I used to teach in Pakistan, and then a long story cut short that now over 10,000 papers are confirming that this virus attacks the brain, and next slides will show you that evidence. So let's start with the number A. If Philip can keep a mouse over that of this virus enters through the nose and causes a viral load in the nasal cavity, maxillary sinus, and not to forget the cribriform plate at the root of the nose, from which it begins possible access to the brain. When I say possible, it was 2020, 13th March 2020 when I said it's a possible route. You will see next slide, coming slides that now proves that. Philip, I want to remain on that slide, please. Okay. That it takes the cribriform plate, okay, to reach the frontal lobe, olfactory bulb, and other adjacent parts in the brain. So this virus is not a respiratory virus alone. It targets almost all the organs of the body, including rare organs like skin and you name it. So we actually researched over this for the last 5 years and proposed two mechanisms that either it's a neuroinflammation that is underlying the cause of the neurotropism, or, and now very certainly established, is that it causes direct injury to the brain as. So when you see the diagram B and B2, you see that the virus attacking neuron actually accumulates plaque, which are resembling amyloid fibrils, which we'll talk in a minute. This is a published paper in which this diagram appeared that there are certain CSF biomarkers which can be seen on the figure B4 that if can be established in the cerebrospinal fluid can possibly lead to a clue towards the neurological involvement of neurological involvement and neurotargeting by the SARS-CoV-2. Now, let me talk about first those lucky individuals who actually could fully eradicate the virus, and then they don't go into a protracted phase of this disease. Otherwise, you can see what's happening in A1 is that patients usually complain brain fog, dizziness, inability to concentrate, okay, shortness of breath, chronic cough, and of that you see many, if not all, of the symptoms, okay, are neurological. So the neurological involvement is now no more in doubt. Over 15,000 papers have confirmed it. And next slide, please. That you can see here the pathogenetic mechanism which were proposed: persistent neuroinflammation, persistence of the SARS-CoV-2 associated spike protein, vascular events like ischemia related to microthrombi, and the most neglected one is placed down where they say ongoing neurological injury or neuronal injury. Now this is the most neglected part of the disease, now getting established that this virus is capable of gaining entry into the cell via not only ACE2 but NRP1 and a list of receptors which have grown to 15 to 16 as I saw the publications last night. Next slide, please. Here you can see the cribriform plate, which you mentioned in that the first paper on 13th March 2020, that possibly this virus is gaining entry to the brain via cribriform plate, which now Philip is moving the mouse over it. You can see the olfactory bulb being affected, and just adjacent to it, over it, okay, is the frontal lobe of the brain. Okay, no wonder why many of them have got cognitive issues related to neuroinflammation. So this virus has got a mechanism of being covered, okay, and hide out in meninges, bone marrow, and the diplo of the skull that we'll talk in a minute. Next slide, please. Here you can see that from the olfactory bulb, okay, after reaching through the transcribrial axis at the root of the nose, the virus can set up three things. It can cause endothelial injury, it can cause hypoxic damage secondary to ischemia or microthrombi, and it can directly cause the damage to the neurons. Okay. Memory loss, ataxia, convulsions, delirium that you can see in the list, loss of cognitive functions. Now, countless people, I mean, like you can't say 16 or 18 million, okay, the count is growing on. And then this cannot be diagnosed conventionally because on MRI and CT scan, it's only diagnosed when there is a atrophy in the gyrus or the sulcitis widening. So hospitalization and seizure, and loss of cognitive functions with cranial nerve palsies are not very uncommon in COVID and long COVID. Especially now, working in this hospital here in Bad Abilene, daily over 27 to 25 to 27 patients we see, and 90% of them are having one or the other form of NeuroCovid. Next slide, please. Now, this is a published paper, okay? The link is possibly given just below the title. You can see that they established microthrombi there. They also found the virus in the midbrain region. You can see in the black and white diagram, it's positive in medulla oblongata. And oh my God, when I used to teach in neurology in my institution, I used to say this medulla oblongata is the center of all cardinal functions of the human body, like cardiovascular system, respiratory system. And just imagine that if it can go seed and kill viruses, neurons there, what could be the outcome? So you see patients with like conscious awareness of the breathing, they can't sleep. All this is related to, again, neurological targeting. Neurofilament staining didn't show any axonal damage, though, the brown diagram, but then the neurons were very deleteriously affected, okay, in staining, this paper showed. Next slide, please. Now coming to what the brain does and what the assistant cells like microglia do there. Okay, so when the virus enters the brain, it sets up a neuroinflammation. The microglia get activated. There is inflammation with secretion of several enzymes. This is an older slide. Okay, neutrophil elastase actually is the mother of them all, which can which can digest the spike protein. When I say digest, don't confuse it. It doesn't convert it into amino acid, individual amino acid, but it converts it into small chop up portions of the spike, which are the so-called fibrils, and these fibrils have been very clearly shown to have a sequence homology with amyloid beta and other forms of neurofilaments, which are very much established, or neuropeptides, which are very established for Alzheimer's. So what are we looking here? Like we are looking at plaque formation, early aging, and Alzheimer's like symptoms in patients who are aged like 55, 53, 47. The youngest one we saw last month was 38. So just imagine, it sets up a cascade of something which could happen in a 90-year-old individual, but then this is a very accelerated phase of aging in the brain. Next slide, please. Okay, here we show that certain drugs like hesperidin, rutin, piperidin actually can block and interfere with cardinal pathways leading to formation of the formation of the amyloid plaques. Also, some drugs like palmidomide, okay, which are derivatives of a very well-known, what you call as a very known drug which caused birth deformities in 1960s. So this actually is a modified form of it. In addition to hesperidin, rutin, and piperidin and myclin, they all have got anti-neuroinflammatory properties. Next slide, please. Now here again, very quickly I will go through it. When we say dysautonomia, or if a person is a junior or a student of neurology knows that autonomic nervous system is a part of nervous system. So NeuroCovid not only manifests by cognitive deficits, but also by several autonomic nervous system dysfunctions and disabilities. So if you see patients who come symptomatically, 90 to 92% of their symptoms can be very clearly ascribed to neurological involvement. Next slide, please. Okay, this one is how it affects the heart, and then some clues towards its action on greater centers of autonomic nervous system and hypothalamus. It can cause a synaptic attack, where it actually mimics and binds neurotransmitter. So the adrenaline and acetylcholine can't exert their effect as a neurotransmitter, leading to dysfunction of sympathetic as well as parasympathetic nervous system parts that you know is very clearly related to the phenomena shown here. When a person stands, then there is no reflex vasoconstriction, so blood cools down and the heart rate goes to 200, 250 sometimes. Next slide, please. Now, this this slide is showing you that how actually chronic inflammation itself can set up serum amyloid-like protein mechanism even without the involvement of the brain. So chronic inflammation actually causes macrophage activation, the interleukins act on liver cells, then they produce the serum amyloid protein, and that actually, with the limited proteolysis, makes the amyloid-associated protein. So patients with long COVID, whether they are a consequence of infection or by adverse effects of vaccine, are showing a very accelerated, what you call as mode of going towards amyloid formation in the brain as well as in the periphery. Next slide, please. So treatment approaches, unlike I've been waiting since 2020 for a drug, okay, that would at least 30% address long COVID and the vaccine adverse effect. Not a single product in the market. They propose Plexovid, and it is failing very bad. You know, Fauci himself got reinfected after having that drug, and it's a full regime. So anti-inflammatory neuroprotective agent in form of nutraceuticals. Stem cell therapy could be an answer. I will reply in the next slide, okay, but then it doesn't stand a very clear chance. You can see the three main targets should be targeting the viral loads, targeting the re-entry mechanism, and the replication. So biclin and hesperidin, you can see down at the bottom right. Okay, we showed in some published papers that these two components, which are part of the combination of 9 chemicals produced by medicinal 9, okay, are very avidly capable of binding to the MPRO and therefore inhibiting the viral replication. So this could be an answer for persistent virus, okay, but one has to take them with the thought that they are not like only plant products. These are very active and important and strong biomolecules. Next slide, please. So neural stem cell therapy sounds very easy, but it's not the case in real, because if the damage is deep inside the brain and not on the superficial gyri and sulci, then approaching that area is so very difficult that sometimes the therapy can kill a person. Next slide, please. That will show you that. So here you can see that if it's accessible to surgeon's hand, then implanting stem cells are possible. But if they affect the deeper portions, like in critical location mentioned here, like in hypothalamus or close to thalamic body or amygdaloid or limbic system, forget about it. I think that implanting a stem cell or a set of stem cells there, the procedure itself is so risky that the operative procedure can kill a person. So where's the answer? What's the answer to NeuroCovid? Until we get any other thing in clarity, my practice here at St. George's clearly shows me that the molecules that we will show you in a minute are very effective, and many of the patients were capable of getting rid of their viral persistence, and even after wax injury have shown substantial recovery of their neurological function. Next slide, please. So here you can see those molecules, biclin, okay, and again quercetin and curcumin, okay, not only published in like conventional journals, you know, but in scientific reports, like at the most bottom of this with the DOIs, they have found to be very effectively addressing the neurological part of NeuroCovid, the brain fog and loss of cognitive functions. Next slide, please. Now these are the molecules. These are just for scientific interest to show their chemical structure, but they have been treated, have been used in even the treatment of non-NeuroCovid related Alzheimer's, and some of the patients with cognitive function and memory recovery clearly shows their potential. Next slide, please. So long COVID with neurological symptoms should have a very structured approach. Consulting a neurologist is the first thing, then individualized therapy is the next thing, which is something we have forgotten in modern age. Not Plexovid is not for everybody, or any other medicine. You have to tailor it to the patient's need. But then these anti-inflammatory neuroprotective agents that you see here, Okay, they address more than 70 to 75% of the patient symptoms in short term, and in long term, we have seen considerable improvement in patients' what you call as well-being and their memory recovery. So current drugs, I'm like very sorry to say, I don't know a single drug in which any biopharma is interested to actually test them in clinical trials for long COVID, in particular the infectious type, or be it the adverse event following the vaccine administration in certain HLA-prone individuals. Next slide, please. I think this is the last one. Yeah. Thank you very much. And if you have got any questions, please feel free to ask. My student always complain I'm a very quick and a fast speaker. I tried to contain it today, but I don't know how much I succeeded. Okay, thanks for your presence and joining us in this conference. Philip always actually facilitates this. I remember the last one we did was a huge success only because we are at the cutting edge of what we tell you. Thank you very much.
Excellent. Thank you. Thank you very much, Manan. I'm going to ask you to hold on while we bring in Shankara and Philip. If you can both unmute your mics, and then I'll allow you to just ask some questions, and I will look out for questions from the audience as well. So let me just take this off here. Manan, so yes, one of you can start, and we'll let you go ahead.
Shankar, you go ahead.
Okay. Yeah, Manan, when you look at neurologic symptoms of COVID, I've seen a lot of acute cases of neurology with COVID, especially with the Omicron variant, but you see very strange patterns of neuropathy. I've had patients suddenly present with senility, dementia, schizophrenia, those kind of conditions, which seem to be easily reversible with conventional allopathic treatment when it's acute in its presentation. Taking the approach that it is an inflammatory process triggered by cytokine release in the brain itself, and seeing that it has such an acute onset, by treating those chemokines and cytokines, you get a good response quickly to them. So it shows direct affectation to the nerve itself. But we also have microvascular issues that create neuropathy. I think those will have long-term manifestations. Myself, I developed microvascular coagulation, which led to seizures. So in your practice, what percentage would you say, or how would you separate those two as to direct injury from the spike protein itself, manifesting as neuropathy, and microvascular problems? Because microvascular problems, I think, will be far more diverse and far more global rather than very targeted. Because I've also seen nerve root problems, specific nerve root problems. So those are, I wouldn't say are microvascular clotting. I'd say it's more targeting by the spike protein itself.
Yeah. So my answer to your question is you are right. Acute COVID is mostly driven in its NeuroCovid part by inflammatory cytokines. The reason it's mostly reversible by a very dedicated therapy to neuroinflammation. But in long COVID, okay, the mechanism doesn't get reversed, but gets established. You know what I mean? The same virus, which was attempting to previously attack human brain by inflammation, now exactly focuses on the neurons. And it is anybody's guess, Shankara, you are a physician like me, after going through the vascular route or transcriptional route, where it would go and establish a host virus relationship is anybody's guess. But unfortunate are the ones that it happens in the medulla. So many deaths have been reported when the brainstem gets affected. But in long term, it actually, and that nerves themselves have got a blood supply. So those microvasculatures, when they get clogged by microthrombi, they go demyelinated. The reason many remyelination therapy and many therapies done with curcumin and resveratrol and also the rutin have improved. So if you can fight that remyelination, long COVID gets addressed a bit. But I'll tell you, in long term, when the infection is there, inflammation is there, but running shows by neuronal damage, direct neuronal damage caused by SARS-CoV-2. You know that a very famous study published by Oxford University authors in which they saw the cortical thinning and deepening of the cell pattern. I mean, we are talking about individuals who go to their jobs and come back. Why the environment part is getting shrunken because of this?
Manan, can I ask you? A lot of people seem to still be struggling to hear. Can you come a little closer to the microphone?
Yeah, sure, I will. So I was saying that the acute phase of COVID is mostly the show is run by neuroinflammation, right? But direct injury, okay, is the ultimate thing which is happening in long COVID patients, because those patients are progressing from neuroinflammation to disability, if not death. You know what I mean? I mean, they come to us in the hospital not by walking and saying I've got a brain fog. They come on wheelchairs, okay? They have got serious what you call as paralytic problems and muscular problems in their body, cranial nerve palsy included. So yes, you are right. The vessels are involved in long term, but acute phase very clearly is run by neuroinflammation.
Yeah, hey, man. Yeah, my experience, what I've noticed here lately is that a lot of affective disorders such as anxiety and depression and people that are totally acting in a different way that I'm not used to seeing. And I would put anxiety probably at the top of that list, where it's almost out of control. And on a structural basis is what I'm seeing. Is this your experience? I'm seeing a lot more brain bleeds, over the last 30 years of my practice. And, and of course, brain strokes, brain stem strokes, and the medulla, etc. Are you seeing the same thing that I'm seeing?
Exactly. I'll tell you, Philip, what's happening is this. You saw the first slide, okay, in which we actually reported it. It was for NeuroCovid, but we clearly proposed in that since the ACE2 is present on the endothelial cell, don't be surprised, okay, if all the body's organs would be affected. And in the brain, especially cerebral circulation, the endothelial cells are studded with ACE2. The reason you see a localized formation of thrombi there, which then releases thromboxane A2 and other chemicals, which actually was a transient spasm. Now, the surprising part is when they do a cerebral arteriography, okay, they don't see anything obstructing it. So I've been very vocal about this for some time, that if you are saying it is a clot or a thrombi, okay, it should be there. You can physically see the occlusion of the blood flow, but you cannot detect prostaglandin and thromboxane A2 mediated vasoconstriction. So that endothelium, when gets damaged, causes spasm. Patient suffer a stroke and a bleed. And when you evaluate him on CAT scan or MRI, you don't find a thrombus or a clot. I mean, this is the surprising part of it. And yes, you are right. You saw one of the slides in which I showed hypothalamus and limbic system. That part, if injured, okay, just Philip, you imagine how much the person is going to suffer psychiatrically and with psychiatric symptoms. So it's very common that they come. Plus, never to forget that if you were jogging and you were running in a park and now you cannot because of an illness, then comes the secondary factor which grips them that I'm never going to be normal. I'm never going to be the same again. So that puts a person into too much depression and anxiety. You're right. You're right.
Manan, coming back to Philip's point, I've had, and I've seen this with other patients, I've had two elderly family members demise post-vaccination with sudden brain bleed, but on scan, it suddenly showed multiple bleeds simultaneously. And I think that was uncommon before, to have someone have multiple bleeds that caused them to demise. Is that something that is unusual and we're seeing more and more since the rollout of the vaccine and COVID?
Yeah, the answer to this question is to find a common denominator, like what's common between the mRNA vaccine and the infection, the spike protein. So, I mean, like when there is infection, the production of the spike protein depends upon the replication of the virus. Okay, in adverse effects, okay, your body keeps on producing it continuously. So these patients are far worse than the infectious one, I'll tell you. Although there are certain specific HLA genotypes who develop the vaccine adverse effect, but if a proper clinical trial was done before the launch of this vaccine, we might be knowing that which HLA fraction should be vaccinated and which should not be. But it never, the time was very short, the vaccine had to come. So the amount of the spike protein that you see in vaccine injury is 10 to 15 times more as compared to that of the infectious variant. And if the common denominator, the spike protein, is actually that high, just imagine that what would they do to end arteries. End arteries have no anastomosis. So once you plug them, the capillaries die and the blood floods out. So yeah, we see a lot of cases, okay, in which the adverse events are causing not non-fatal, what you call as events, but mostly fatal and paralytic events. Yeah.
And just coming in as a final question here, Manan, when you
Please.
think about what you are looking at from a research point of view,
Mhm.
similar to what we have, one of the big challenges with neurology is once damage is done, there are very little ways to reverse it. Would you say that if nothing is done at this point, for example, to try and mitigate damage, that the consequences down the line will be irreversible?
I remember one of the invitation I got from Nature Reviews Neurology. Okay, the conclusion line was saying that if you don't treat these disabilities, they would turn into deaths. So this is the line that you are saying. Neurons, skeletal muscle, cardiac muscle, and adrenal medulla, they never regenerate. Damage once is damage forever. Neuron would not regenerate. So if you don't address these issues now, the number of the people that you are seeing now disabled, okay, you would be seeing that they would be vanishing, okay. The certainty of chronic disability would lead to deaths. So I don't know where is the investment. Why don't I see any investment or funding for this type of disease, which is crippling individuals? I know chartered accountants, bank manager, university professors, okay, they cannot get out of their bed, okay. What's happening here? I mean, like, come on, give me a break. I mean, this is psychosomatic. I would like those who say psychosomatic to this disease themselves should have a brain scan or an MRI, because the person would not leave a carrier in the midst of his shining times, okay, and lay down in the bed and pretend that, oh my God, I've got depression or something. There is a serious problem here. I mean, like, with doctors, I mean, like, my peers, some of them, okay, say, oh, show me in the book, where is long COVID? Why can't I see in the book? What are the signs and symptoms? So when will appear in the book, okay, and then you will read it, the mortality will be in millions, okay. So why we are waiting for those traumatic times and bad times to come? Why not address the disease upfront now? So you see now they have got a vaccine adverse effect reporting system and other compensation going on. I mean, like, we are getting late here. I mean, like, why are we not taking the disease upfront? That yes, this is a disease, recognize it, fund it, treat the patient. These patients were contributing as taxpayers, okay. Their money is actually making government stand on their feet, okay. Now when they are sick, government turns the back. I mean, like, I don't understand. This even doesn't happen in my country. We have got free clinics, okay. We treat patients for free, even if they have got no medicines. So my heart bleeds, okay. And I'm like not pretending or acting here, and I'm not very fond of getting what you call as an award here. No, this is a serious problem. Governments have to listen. Whosoever is listening this podcast, our recording, when it goes, please try to understand. These people served you. Okay, this is now the time to serve them. If you don't do your part, history will write some very bad things for you. Okay, which you would never imagine now sitting on the official chair. Okay, they will not remember you as a good leader. So that type of involvement is needed, Philip and Shankara. I mean, I don't see that involvement. Sorry.
Hey,
Wonderful.
Philip, can I ask a quick question?
Yeah, go ahead, Philip.
Yeah. Hey, when you were talking about stem cells, has your group or you tried any? We're using a technique called soft wave, which is a hydraulic sound wave. Have y'all used any of that to produce stem cells in the brain?
In this case, Philip, we need to have even if it's a neuron or a cardiac myocyte, we have to have an autologous type of mechanism of stem cells. Okay, otherwise you actually plant a stem cell and then wait for a rejection if it's not the same HLA, or the match. So nowadays we are going towards that step. But then you saw one of the presentation which was showing that if the defect is in medulla oblongata, midbrain, even before the surgeon's hand reaches that part, okay, the patient is dead for good. It's not a child's play to play in those areas. Yeah, cortical parts, yeah, occipital lobe, temporal lobe, I mean parietal lobe, surface gyri, you can implant cells there. But implanting it in amygdaloid nucleus, and limbic system, the central core parts of the brain, medulla oblongata, it's not happening in the next 15 years.
Wonderful. All right, we're going to have to move on now. Thank you very much for that, and we'll thank you, Mannan, and we'll certainly talk in the round table. And so, without further ado, we'll get straight into our next presentation, which will be with Kevin. So, Kevin, I'll bring up your slides, and we'll let you get straight on with your presentation.
Okay,
One second.
Yep, so as I said earlier, I'm a systems neuroscientist. Just as a full disclosure, I'm currently running my own company looking into the effects of COVID. We didn't establish the company in 2017 to do that, but that's where we are at the moment. Hamsters are our model system. What I'm going to do in this talk is basically give some of the history of what happened, why are we here, dealing with, as Mannan was saying, a tsunami of long-term injured, and what is the probable cause for this injury. And as you can see from the title, I spend a lot of time looking at amyloidogenic mechanisms. So just to give people some background, you can go to ResearchGate, you can find all my publications there from when I was working in academic environments. Most of them are free to read, and again, this is the company I founded, and I'll get to this at the end for those that perhaps want to do some of the testing that we're currently running. Okay, so I'm going to start, as usual. I always put this slide in. I was one of the first to be talking about SARS as a synthetic chimera. As evidence began to emerge over the last 5 years, we're very confident that it's a consequence of DoD funding, along with their medical countermeasures, the vaccines, which were essentially mandated and forced onto the population at large. These all constitute crimes against humanity. There were a number of features that were immediately standout. There was overlap with HIV, and if you were keyed into the proteomics, you could know that there were amyloidogenic sequences, and HIV stands at the very heart of the research that has gone into making this type of chimeric next-gen biowarfare agent. So just to try and give a broader, unified picture of what we're dealing with in my field, in the neurosciences, around it's a good 12, 13 years ago now, the conceptual leap was made that there was a unifying mechanism between a number of neurodegenerative disorders, Alzheimer's, Parkinson's, etc., and this all revolve around amyloidogenic mechanisms, which means that you get a oligomerization and misfolding of proteins, in this instance in the brain, but they can happen systemically as well. I'm going to use this presentation to maybe talk a little bit more systemically as to what we see with respect to COVID and spike protein related amyloid pathologies. So it's just a quick table just to inculcate the fact that there's many proteins. This is just scratching the surface with respect to proteins that can become amyloidogenic. And like I say, my area of expertise was Parkinson's, modeling Parkinson's, etc. But the field is very wide and very broad, and we're beginning to, particularly in the last 5 to 10 years, really expand our knowledge on how these misfolded proteins lead to disease states. Now, As COVID was beginning to make its impact globally, there have been a number of key researchers who have published some of the what I consider the most important work, which was to identify these amyloidogenic sequences in the spike protein, not just in the spike protein actually, but I'd save that for another talk. But many of these sequences, short 20 amino acid long codes or epitopes, were were unique in their presentation. They hadn't been seen before, and they you could do computational analysis to try and work out if they were amyloidogenic. Now, the same group published that, well, it still sits on preprint server, and what they showed was that there was a 20 amino acid sequence in the spike protein that had a predilection to catalyze the prion protein, which is the one associated with Creutzfeldt-Jakob disease, things like BSE and McCairn. And this 20 amino acid sequence had the most avid reaction out of all the reactive seeds that they looked at. And when I'm talking about seeds, I'm talking about these 20 amino acid long epitopes that they would take and test to see how long it took to cause misfolding and the characteristics that we associate with diseases like Creutzfeldt. So this was very concerning. I've spent a lot of time trying to replicate a lot of this data, but the important thing that I'd like to I'd like to ensure that people understand this critical point, which is that when they made the countermeasures, in this instance, the mRNA vaccines, they copied sequence by sequence, amino acid by amino acid, and the vaccine spike protein had exactly the same amyloidogenic footprint as the spike protein from the natural, well, not natural, but the synthetic virus that was circulating globally. Now, this is very concerning from a proteomic standpoint, and just to reiterate a bit of history, which this first paper from Nystrom and Hammerstrom, when it was put on a preprint server, they had a section in there basically saying that it seems an incredibly bad idea to make these vaccines have the exact same epitope, amyloidogenic epitopes, as part of their protein structure, and they were forced to take that out for the publication. And that was that's a indicator of just the atmosphere that we were working in 5 years ago, as scientists tried to raise the issues around controversial, maybe not so controversial these days, but again, the synthetic nature of the virus and the potential fallout from using gene transfection technologies to express the peptide in the body. So let's move on. So do we have any evidence of the mRNA vaccines causing a amyloidogenic reaction in the body? And this, I recommend this paper to everyone, which is this was an individual who was scheduled to have a PET scan because they were looking for a diagnosis as to why he was showing signs of dementia. But the day before he had his scan, he actually received a Pfizer shot, and in the left-hand corner you can see in his arm the ligand that binds to misfolded amyloid beta was tagging the area around where the injection went into his arm. So this was some of the first evidence coming out that exposure to the spike protein could cause these amyloid cascades. And if you get into the biology, they're very, very difficult to treat, and essentially are irreversible diseases. And you know, just with some history, you could look at the causes. So we had a lot of excess death during the period of COVID. Now, what caused, or what was the nature of why there was this spike in excess deaths? And Alzheimer's and dementia and circulatory diseases were the primary driver of excess death during the first, well, you could even say that it's still ongoing, as we have excess death running at around 10%. It's certainly high here in Japan. And we had recently, just last year, nationwide studies coming from, in this case it was South Korea, where they had a very strict vaccine regimen. And they published that there seems to be an association, and again, this linkage between exposure to spike protein and early onset Alzheimer's and cognitive impairment. And so this spectrum of symptoms could be coming from many different sources, but some of the most important work, I would say, with respect to looking at the pathology of spike protein exposure has come from Lucia Prestoris in South Africa and Douglas Cowell. And what they've shown is that fibrinogen, when exposed to spike protein, becomes amyloidogenic and resistant to breakdown, and it begins to form microclots. And these microclots, being amyloid in nature, are detectable relatively easily if you apply the right techniques. But the principle that you're having, or the body is under sustained microclotting, amyloidogenic microclotting cascades, which lead to downstream knock-on effects. And you know, there's a principle in amyloidogenic diseases around cross-seeding, and they actually did early work which showed that microclots are actually primarily fibrinogen, but you find other misfolded proteins in them as well. So pointing to the complexity of what we deal with as a causal factor in why we see long COVID, why we see the impact on brain function, etc. And I would say that the chronic depletion of oxygen as the blood begins to fill with these microclots is a major driver of these symptoms. And yeah, we just done this. And so as we sort of, I'm going to move towards looking at some of the extreme amyloid expressions. These are the calamari-like clots, which were found by Richard Hirschman. But I want people to sort of look at this, the title of this paper, which is that there is a known interaction between fibrinogen and scrapie prion protein. And fibrinogen basically acts as a safety mechanism in the body and can potentially bind scrapie prion protein. And I want people to think about all these converging mechanisms as we look at the nature of the amyloid clots that have come from cadavers. Another paper, just quickly, from the Hammerstrom lab again. And what they show in this paper is that there are, again, these short epitopes, and they bind specifically to the fibrin and are part of the initial causal mechanism to make fibrinogen turn into this amyloidogenic form. And so this leads to clotting disorders. This is something we see in the hospital that I collaborate with here in Japan. There's a patient that received Pfizer and basically had a severe coagulopathy of the arm, eventually resulting in its loss. And we'll skip the movies, and we'll get to the calamari clots that have been retrieved from cadavers. Now, what's the causal agent for the emergence of this phenomenon? It's very, very difficult to pin down at this moment, and there's a lot of controversy around this particular discovery. And I'm just going to go through what we've seen in the lab and maybe dispel some of the more egregious should we say, I don't want to say conspiracies, but some of the potential narratives that are being pushed out with respect to what these are. And you're basically looking at a calamari clot that's fixed in argon or formalin. So the argon is on the left and formalin on the right. And we're basically just going to go down in scale, looking at the nature of what this amyloidogenic fibrin that is that's appearing and emergent in scale, orders of magnitude larger than the microclots that have been recognized within the academic literature. And we have a problem at the moment that the academic side does not want to put the effort in to look at this phenomenon. And so it's private labs like mine which are doing the work. So there's one of the calamari clots, just for reference. That's 2 hypodermic needles holding it down. And we have done PCR. This is work done by Kevin McCairn, and we can confirm that it is human tissue in origin. It's not something, it's not a fake being pushed out from, I don't know, an abattoir or someone trying to spoof the population. And I'll quickly skip through this and get to the gross anatomy. And here's a close-up of the calamari clot. You can see that there are fissure and sulci in there that's sort of consistent with it being embedded within the vascular system and being compressed against the side. We can cut through it there. It seems somewhat solid at this scale, but as we cut down using techniques for microscopy, microtome, and we can, in this instance, we're going to look at 3 slices from the argon fixed specimens. And when you look at it at low power, you can see that it's not a solid tissue. It has a mesh-like network, which becomes more apparent as we increase the magnification. And when we're looking for amyloidogenic proteins, there are certain markers that we want to see, so we can begin to tick boxes that say it conforms to an amyloid-like property. And one of the first things that we see is that even without staining, these large calamari clots are autofluorescing, and microclots have been confirmed by Pistorius and Kell as being autofluorescing. And when you add dyes that are specific for misfolding of protein, which is on the left-hand side, you can see that it lights brighter green, and you can see a polka dot appearance to the tissue. And again, just increasing in magnification, and you can see the close-up of the polka dots and the brightness, the avid take-up of the dye, thioflavin, which is indicative of misfolded protein. So now we're going to use scanning electron microscopy to go down in scale. We want to get a understanding for the nano scale protein and peptide structure. So here we're looking at 25 times, 250 times. So we're going beyond the limits of light microscope. Here we can see the section, and just to help orientate you, we're basically at this scale looking at one of these fiber meshes when we get down to this scale, 1,000 times. And then if we look at it at 5,000 times, what we see is a very twisted and nodular appearance to the protein. And that's very, very indicative of amyloidogenic presentation. And here you can see, I encourage everyone to read this study, Fibrin Drives Thromboinflammation and Neuropathology in COVID-19. And here they've basically, on the left-hand side in the bottom corner, you can see normal fibrin. It has a sort of smooth spaghetti-like experience. Here they exposed it to spike protein, and now you can see that it's twisting and has the nodular appearance that we see in this image here. I'll skip through this. It's basically just elemental analysis. People have spoken about tin being present. I didn't find any of these sort of metals in the tissue, just protein elements you would expect to see with normal proteins. Skip through that. And now there are mechanisms or techniques that we can use to try to confirm if there is a amyloidogenic core. So we're looking at the so we saw the large twisted fibril at 5,000 times, and there's a technique called Raman spectroscopy where you expect to see a peak at a particular wavelength that's thought to be indicative of these amyloidogenic misfolding properties. Now, in the canonical proteins, that peak is around 1680. In the calamari clots, it's shifted a little bit to a higher frequency. There can be a number of reasons as to why that occurs. So it's not directly comparable to the known or established amyloid proteins. And some of the things we've been doing in the lab is techniques called RT-Quick. We have seen some reaction and conversion to scrapie prion, just some of the raw data. Now, this bar chart is representative. So there are many tests being done, but on the end column here, we took a seed amount of the calamari clot, and in this instance challenged it against donor plasma. And what we see is that after 7.5 hours, we get an exponential rise in fluorescence, which is again using thioflavin, indicative of a amyloidogenic cascade occurring in the plasma. Now, a lot of people ask me about treatments, etc. In the lab, we've only seen one effective treatment. This is just the raw data. Basically, the red bounding box is untreated, the green is treated, and in this instance, it's been treated with stem cell growth factors derived from tissue pulp from children's teeth, basically. So it's an ethical source of stem cells. And just to quickly run through it, it's a lot of raw data, and I want to wrap up. I know we're at the end of time for the discussion, but basically every concentration of stem cell growth factor inhibited the amyloidogenic cascades. And I'll quickly finish with what we're able to do in the lab. We can look for and detect microclots from dried blood samples. People have been vaccinated. This is someone who received Moderna vaccine, and you can see these long amyloid fibrillar-like forms, and here we are stained with thioflavin. And I could finish here, but if you indulge me a little, there's some concern about the effect of spike protein and COVID, etc. What's the impact with respect to transmission through pregnancies? There's a lot of data out there that there has been significant harm, and is there transfer to children when the parent has received vaccine. And I received a sample from a young boy, 3 years old. His parent or his mother was vaccinated at 32 and 34 weeks. He was basically born premature. A week later, arrived. He wasn't alive when they took him out. They resuscitated him, and I was sent samples of his blood, and standard sort of testing. We can find amyloidogenic signature, these fibrilla-like forms, and here's some scanning electron microscopy of the same form as well. So we do have evidence that this amyloidogenic prion-like disease cascade may be passing to the next generation in parents that have been exposed. Now, I can't discount that the child was exposed to COVID as well, but with respect to the circumstances of his birth, I think signals like this need to be taken into account. And yeah, I'll finish there. I'll just add that if you want to have the blood test, get an idea of the amyloid burden, go to my website, click on protocol and sending blood samples, and I'll check it for you.
Excellent. Thank you very much, Kevin. Wonderful. And what I'll do is I'll add in Shankara and Philip as they go through their questions, and I'll probably look through the audience comments and see if I can add anything else that could be relevant as well. So I'll let you go on. I don't know who wants to go first.
I'll take it if that's okay, Shankara.
Yeah.
Hey, Kevin, I know your research, you found that one of those hits on your RT Quick showed some pryogenic type of disease. Can you explain to the audience more? We know there's multiple types of amyloid, right?
Mm-hmm.
Pryogenic is a little bit more of a beast-like situation to where, you know, that you can't kind of stop it. And it's more of a sort of like an infectious type of process. Could you explain to the audience, kind of like in a layman's term, why that is important and why that takes it to another level of depravity?
Yeah, so this comes down to exposure through multiple pathways, and one I'm particularly concerned about is blood products and people that might be carrying these amyloidogenic clots. I don't think there's any systematic check for them at the moment. But in a sense, what happens because of the nature of the protein and its misfolding, it basically acts as you can think of it as like a magnet, but as it approaches a protein of similar species, if you like, they stick together, and that sticking together causes the misfolding, and it makes it more difficult for the body's enzymes to break it down, and it will build up systemically to the point where if the numbers are to be believed with respect to the calamari clots and their frequency, that they go to an extreme size. Now, you know, I often get asked why the size differential between what we're seeing or what Richard Hirschman has found relative to the language that we would use, scientific language around amyloids and neurodegenerative disorders, and all the fibrillar forms that you would see with Parkinson's, etc., are very, very small. They're intracellular or just parked extracellular when as the disease progresses, and so it's the boundary condition of the cell membrane which constrains the size and the form. Now, in this instance, the boundary condition for these amyloidogenic microclots is essentially the circulatory system, and so there's no theoretical reason as to why they would stop growing, and the length, essentially, you can read into there are many papers out there as to the actual length that they could grow to. Again, it's a complex disease mechanism, there has been enormous amounts of exposure. 5 billion people have been vaccinated, on average 2 doses, and we don't know the degree of penetrance into the population with respect to this amyloidogenic disease-causing cascade. And it doesn't just come from the vaccine, it does come from COVID as well. When I'm looking at the microclotting under the microscope, there's no difference. I can't tell them apart. If you didn't tell me what the patient status was, all I could tell you was that there's microclotting, fibrillariform microclotting. They look very similar. I hope that answered the question.
Kevin, following on what Philip has mentioned, I see specific phenotypes of patients that are more prone to developing microvascular clotting from the vaccine and the virus itself. So I see different presentations, but they seem to have a phenotypic propensity. Some patients with Omicron will develop respiratory symptoms. Some patients are more prone to develop bowel symptoms, and some patients more prone to the microvascular clotting, which seems to be associated with neuropathy or neurologic symptoms. Would you say that those phenotypes more prone to developing microvascular clots will also be those phenotypes to have more amyloidogenic potential?
Yes, I would think so. My issue is that I'm not a frontline MD treating patients. I'm doing the basic research in the background, trying to do the in-depth work, trying to extract answers out here. So yes, I have no problem doubting that there would be different cohorts that are susceptible. You see that with any sort of disease. Pick your poison in this instance. And why is that? Genomics, environment, again, it's the same boilerplate answers that you would usually get. I think the important issue here is that we have to recognize that there is this clotting, amyloidogenic clotting in extremis that is not being addressed by the institutions which are supposed to be there to be protecting the public. They're being given very privileged positions, and they have that access to the equipment to be able to answer these questions quickly, and they haven't done it. And you have to ask why. And the only reason that I can come up with is just that there's so many institutions, entities, individuals who are basically culpable for this mass exposure and this potential mass casualty event that they've inflicted on the global population. So no one, there's no motivation to go and investigate this, and it may as well stay as just a microclot phenomenon, right? That's enough for some sort of diagnostics, but are they really getting to the extreme end? And how many people and patients are walking around with these large clots? And it's something that perhaps Philip can speak to with his experience.
Just before Philip, I just wanted to grab something from the audience here, Kevin, and this is a question I had as well. I'll let you go afterwards, and it is here. Is it possible that cerebral amyloid angiopathy could be triggered by the spike protein?
Yes, very much so. I think that's been demonstrated in the literature now. All manner of vascular assault is ongoing, and it's happening at multiple organ systems, including the brain. Again, how to catch and identify the people who are going to be impacted by this reaction. You can do the type of testing that I'm doing. It's relatively quick, it's cheap, it can give you some idea, but then you have to think about treatments. And in vitro studies, the only thing that I've seen that had any meaningful impact was stem cells. I had Mannan mentioning those as well, but we're still we're very much behind where we should be with respect to understanding this phenomenon, with respect to the resources that are available to these institutions that are essentially ignoring this phenomenon.
And Philip, could you ask the last question before we move on to the next speaker?
Yes, about the clots. He was speaking about Richard Hirschman and the cadaver clots. Well, we're seeing these in live in people that are still alive. Have colleagues that are interventionalists, right, whether radiologists or cardiologists, and they are pulling out these clots, these big white clots, these non-normal amyloid type clots, and they've been doing it for the last several years. So, and I was just in the hospital recently visiting, and he showed me some recent ones, and he says it's occurring on quite a frequent basis. So yeah, they're occurring before post-mortem. They're ante-mortem, they're pre-mortem, and in my opinion, we're seeing these microclots that Kevin's picking up, and I believe that these are forming into the macroclots that will eventually, unfortunately, come to someone's demise or loss of a limb, etc., stroke, whatever.
And it's not just vascular disease. Misfolding proteins are intricately involved in the generation of cancer and tumors, and that's something that we see. In Japan, the sustained all-cause mortality seems to be coming from cancer. That's what we see in the clinic, unusual cancers in this instance. And I would say that there's evidence out there that that's occurring in the US and other countries as well. And what Dr. Philip talks about, which is the autoimmune issues and the follow-on cascades from that, all of it's converging and causing a disease burden that, again, is I think their strategy is just to say, okay, we're gonna receive these patients into the system starts treating them, and we don't really discuss why they've ended up needing hospital care or medical care in this instance.
Wonderful. Thank you all. And we're going to be able to deal with some of these a bit more closely in the roundtable shortly after this. But let's hear our third and last speaker, and then we will move on from there. So if you just hang fire as I bring in now Dr. Diane Counce. So Diane, I will let you go straight into your presentation, and I will bring up your slides and I'll move them along as you require.
All right, great. Yeah, so the science is great, and this is something that I'm going to talk about as far as what I see clinically. So next slide. And I had a lady, Mary, 61 years old, who did complain about having new onset headaches in 2022. She was diagnosed by an outside neurologist as having migraines. And she did have some migrainous components. She had throbbing pain, light, noise, sensitivity, worsened with exertion. But there's just a little bit something different. One, her age, that should immediately raise a red flag for people. Migraines are typically, teenage years to about the age of 55. After the age of 55, you need to start really thinking about something else. And also, these were continuous headaches. They were occurring 24 hours a day. They were there when she woke up, when she went to bed at night. And they were also unresponsive to typical migraine medications. So she did have an outside MRI, carotid Doppler, both normal. Someone thought about sleep study, which is reasonable. Someone has sleep apnea, they can have a different type of headache, but it did not make any change in her headache when she used BiPAP for about 9 months. And then she was referred to me May of 2024. And I did get the history that, by the way, she did get 3 Moderna shots, and then she had 2, possibly 3 COVID infections. And I think it's important to kind of get the dosing of spike protein, be it from either the infection or vaccines. I think that puts a more severe slant on some of these patients' symptoms. Next slide. So not only just headache, but now I really dive into patients' review systems. It used to be something that we just think, oh, insurance requires us to ask, all of these things. But now it plays a really, really important role as far as trying to figure out, do they have a spikeopathy? And so her in particular, she had weight gain, shortness of breath. She couldn't go upstairs 2-3 times. She had sweating. She was feeling hot all the time, cold, restless sleep. And then she had some negative symptoms as listed below. Checked some labs. She did have some normal labs, which was a little bit surprising because she didn't have very many inflammatory type markers. But these markers aren't going to be 100% when you are trying to diagnose long COVID or long vax. So just kind of be careful of that. Since I was pretty convinced that this is what she was having, I did put her on ivermectin and then several supplements kind of targeting cerebral blood flow, inflammation, mast cell with the famotidine and the cetirizine, probiotics, and then nattokinase because she had shortness of breath. I was thinking, could there be some microclots that were causing some problems? Next slide. Oh, go ahead, next slide. So at her 2-month follow-up, after we put her on all those supplements and the ivermectin, she was actually headache-free within 3 weeks. She still had some problems with temporal regulation and weight gain. So we talked about putting her on a gluten-free diet based on her sensitivity to gluten, intermittent fasting, and then we really kind of stressed, starting some of the other supplements that we had found abnormal labs for. Now, on her initial physical exam, she did have some neuropathy, and that correlated with her sweating and hot sensation that she was feeling. And sure enough, on nerve connection studies, she had a sensory neuropathy. And we have a test called CMAT, and it actually looks at autonomic nervous system abnormalities. And so to me, it takes the place of a tilt table test without having to do one of those testing. And it's really kind of nice. It shows not only dysautonomia, but presence of small fiber neuropathy. And that is one of the most common things that we're actually seeing in people post-COVID and post-vaccine. She'd only had 2 or 3 headaches, from the month before, but they were very easily treated. And by starting the intermittent fasting, gluten-free, vitamins, and so forth, she actually had a 10-pound weight loss. All right, next slide. This is a gentleman, 37 years old. He had 2 Moderna shots, and then he had COVID January 23, developed fatigue and respiratory symptoms February 2023. Started seeing one of our local COVID literate physicians, Dr. Jordan Vaughn, and he placed him on several supplements, and Eliquis, aspirin, and ivermectin because he did have some microclotting based on his immunofluorescence microscopy that he does. And he was able to decrease it from 3.5 to 1, being placed on those supplements and medications. And sure enough, his respiratory symptoms did improve. However, in May of 2023, he developed his first episode of unconsciousness and unresponsiveness. He lost his brother. And of course, these were witnessed by his brother. And brother said that he makes a weird sound before these start, and he's still asleep. He's breathing deeply. And then, as he's coming out of sleep, they're noticing some strange, activity, not really tonic-clonic activity that you'd see with a generalized convulsion. But then he'd develop muscle soreness and fatigue for the next day to, 72 hours. And I've always been told that 3 things that come out of sleep as far as movement would be either the parasomnias, periodic limits of sleep, or seizures. He had a total of 15 events since May of 2023. He was seen by another neurologist and was basically told it wasn't neurological because all the studies were normal. Routine EEG was normal, which can happen 50% of the time that someone does have epilepsy. And so he is, of course, given the diagnosis of functional neurological disorder. And unfortunately, that diagnosis has increased by 60% since the start of COVID and the vaccine rollout. Next slide. We did get a NeuroQuant, and so over on the right-hand side, you can see the breakdown in the numbers. So blue means that the area that is measured by AI technology is 95% or higher, and pink is 5% or less. And so you can, and I take that into looking as the cells in that area are plump. You won't necessarily see it on a routine MRI, but the cells are kind of plump, maybe from inflammation bringing in fluid. And so you can see that he had sort of diffuse neuroinflammation with the temporal lobe, parietal lobe, frontal lobe showing a lot of blue in those areas. He did also have a 1 percentile in the cerebral white matter over on the left-hand side that's there in pink. So, of course, this was discussed in detail, and in particular, the hippocampus had a lot of blue. So, could there have been, a lot of inflammation, aggravating a very epileptogenic area, of the brain, the hippocampus. So, given that he had stereotypical paroxysmal events that happened out of sleep, even with a routine EEG being normal, they wanted to go ahead and start a seizure medication. So, lacosamide was started. I also started medicinals, which is molluscum, I think is what they call it in Europe. And I did do an ambulatory video EEG for 5 days. That was still normal, but 95% of patients will have an abnormality. That means 5% will not on the ambulatory EEG. So you can still miss seizure patients. And then as a follow-up 3 months later, he's had no further events. And we are going to plan on doing another NeuroQuant to see if we have changed the inflammation on this gentleman. Next slide. So this is a patient on the right-hand side. She's 70 years old, came to me complaining about memory trouble. And I bring in to Dr. Michael Nell's information on the indoctrinated brain, which I thought theory into practice. He talked about the spike protein in red number 1 landing on receptors, including the TLR4 receptors, part of the neurons. And then that will cause an escalation of cellular mechanics starting and then spewing out these cytokines over in the red circles, interleukins, TNF-alpha, and so forth. And with enough inflammation, then you can start having damage to the hippocampus as far as neurogenesis. And you can still, continue to make connections in the hippocampus, and that's why we always promote, social interaction, reading, any kind of word puzzles, and so forth. You can still create connections within your hippocampus, but with so much inflammation, you will actually start to block this. Over the right-hand side, you can see the NeuroQuant actually kind of makes sense. You have all this blue in several areas, including the temporal lobe, the parietal, frontal, occipital, even whole brain on the left-hand side of that NeuroQuant, so at the very top. And then the cortical gray matter, all of that is greater than 95th percentile. And look at the poor hippocampus in pink. It's at the 1 percentile. So this is something that I think Dr. Nels was kind of alluding to, that too much inflammation will then start damaging the hippocampus. All right, next slide. This is an interesting gentleman. He was a trucker, and so he developed left-sided weakness and aphasia. He was actually able to call his wife, but she couldn't understand him on the phone because of his aphasia. So they found him by GPS, and he had 3 normal MRIs, and he was basically told there's nothing wrong with your brain, although he completely looked like he had a stroke by all practical purposes. So I saw him, did a NeuroQuant, and you can see on the right-hand side that a sea of blue, lots and lots of inflammation. And granted, prior to this, he'd had a couple and a couple doses of infection. And so, but pay attention to the one pink area, the right pallidum. And so with that, that's a area that controls the motor. And with that, I doubled his nattokinase. I had him on ivermectin and a lot of other supplements, but I doubled his nattokinase. And within about a week, all of his symptoms went away. He was no longer weak on the left side, and his speech actually returned. So that's how I kind of used that, NeuroQuant in that particular case. Next slide. All right, so Randall, I saw him previously as a migraine patient in 2018, and then he returned to me February 2025 But this time his symptoms were different. And now he was having a pressure in his head and the eyes, almost blacking out, feeling faint. He had a sensation of falling, vertigo, brain fog, and he'd have these in episodes. And during one of his episodes, he almost drove off a 3-foot, well, he did drive off a 3-foot retention wall during one of the episodes. He had already had a previous MRA of the neck, carotid Dopplers, ophthalmology evaluation, and then a CTV looking at the veins. The radiologist read it as normal. He took it to a neurosurgeon in New Jersey. I'm not sure how he found that neurosurgeon. The neurosurgeon felt that he had an undeveloped transverse sinus and thought he could stent it. He was suspicious of intracranial, idiopathic intracranial hypertension. So, I saw him February 2025 with that workup already done. Thought, well, could this be a strange migraine variant? Could it be the IIH? Because he did have improvement of symptoms after changing his diet, losing weight. Diamox helped, but he had to stop it because of side effects. He did find it, we did find him to have some peripheral neuropathy on exam. Next slide. So I checked labs, put him on some naproxen, 50%. I was trying to figure out what is going on. And then he followed up in June of 2025. And that's when I thought, what is possibly going on? What did you do as far as COVID shots and so forth? So he had basically 5 doses. The last one was 2024. He had a really bad reaction to his first shot in 2021. It included brain fog, pressure for 4 days. He couldn't even function. He couldn't go to work. And lo and behold, it was similar to his current symptoms. And then after he had COVID in 2024, symptoms really, really worsened. So I put him on ivermectin, low-dose naltrexone. I stopped his statin. And that's something that I do a lot of because the brain actually needs cholesterol to feed on. I think statins are, not a good product, but that's a whole nother talk in it by itself. And then of course I checked NeuroQuant in the COVID labs, and the MRI that I had, part of the NeuroQuant, I always have to get a baseline MRI. I wasn't quite impressed with the transverse sinus abnormality that the New Jersey surgeon was seeing. Now, granted, I did have the CTV, and so, but this was just a, one shot, looking at the right and left transverse sinus, but I was unimpressed with need for a stent. With it. It was a little lopsided, but I didn't see a significant stenosis. All right, next slide. So after putting him on low-dose naltrexone and ivermectin, he actually got a little worse, which is not unusual. There's a Hertzheimer response where all of a sudden you sort of get an unleashing of these toxins, but then he got better. And then at follow-up, he wasn't quite where he needed to be, so I increased his low-dose naltrexone, added the medicinals. And over on the right-hand side, you can kind of see, the NeuroQuant that I had. And again, a lot of neuroinflammation, whole brain and cortical gray matter on the upper left-hand area. And then on the right side, every single, lobe was affected. Fortunately, he didn't have any areas of atrophy, which would have been in pink. So when I saw him in follow-up in August, it was kind of funny because I walked in and his eyes were really big and he was kind of leaned up and he goes, "What is going on?" And he said 80 to 90% of his symptoms had improved. He had no more pressure, no vertigo, blurred vision, brain fog, sensation of falling. He is now able to exercise 4 days per week. And he had not been able to do that. Every time he exercised, he'd go into one of these spells. All right, next slide. Now, this unfortunately is a lady that 59-year-old. She's a CRNA. She both worked at the hospital and also taught. And I think this is something that Kevin might have alerted to, that these are brains that are very, very damaged. She has a sea of red. There's some inflammation. But most of it is red. And these are brains that I think are going to have very, very poor prognosis until we can figure out how we can prevent this. And you can see that the UK Biobank actually did neuroimaging study. Patients had MRIs inadvertently before COVID hit, and then after COVID, and they did have all evidence of COVID at the time. This was before the vaccines actually came out. And they were able to allude that instead of a 3-year aging process, it looked as if their brains had aged 10 years. So this is what we are up against, and we don't want to see seas of red on this patient. By the way, the CRNA actually had to quit her job at the hospital. She's still trying to teach, although she's having a very, very hard time with recall of her information that she was trying to teach. Next slide. Now this is a different, and it's a little bit hard to read. I couldn't make it bigger. So you can actually get either TBA, which is total brain atrophy protocol, or a NeuroQuant protocol. And 52-year-old science teacher, and she was having a little bit of memory troubles. Of course, she was forced to get the shots. She's had a couple of COVID infections. And so I did do the NeuroQuant. On the right-hand side, you can see the two far bars are the prior, and then on the left-hand side are the current. And so this dementia protocol, they hone in specifically on the hippocampi. And you can see on the very top line, the hippocampi prior was at 7th percentile on the far right. And then 2 bars over, you can actually see 8 months later, it actually increased to 9. Now, could that be, within the normal variance? Possibly. And then at the very bottom row, you can see the hippocampal occupancy score. Now, that is the space of which the hippocampi reside. So, of course, people have smaller spaces and larger spaces. So, in April 2024, her hippocampal occupancy score is 21% on the far right. And then 8 months later in December, when I repeated it, it went up to 30. Her symptoms have improved with my treatments that I've been giving her. And then the other interesting thing is the whole brain, which is about 5 lines down from the top. You can kind of see on the prior, it was actually 84 percentile. And then 2 over on the current in December, you can see it actually dropped to 68 percentile. And so I was kind of thinking, okay, am I doing something that's, the whole brain now is getting smaller? I don't think so. I think we're actually decreasing her neuroinflammation overall to actually get that decrease of the whole brain, but the increase in the hippocampi and the hippocampal occupancy score. So that's how I interpreted the NeuroQuant with that. And of course, I do have of NeuroQuant does mean it's volume. I talk about density. I'm kind of curious. I kind of ran across Dr. Diane Counce. He actually has a scan that he's calling the biometrics neuro MRI, and it actually includes almost a DTI, functional density scanning, volume scanning, all in one. And I think that would be very, very, exciting to be able to utilize those in clinic. And then last slide. This is going to be the hardest thing, and this is what the other two speakers have alluded to. We have a long road to go. We don't have answers yet for again the sea of red, dementia, Alzheimer's, Parkinson's, ALS. I do have to say that I'm seeing these almost like people are complaining about the turbo cancers. I'm seeing turbo Parkinson's. I have patients that wake up one day and they have Parkinson's as if they have had it for 5 years. Whereas before it'd start, with just maybe a little tremor in the thumb, and then gradually over a couple of years get worse and worse. This is just frank Parkinson's from day one. It looks like they've had it for years. Same thing with dementia. We're having, very, very fast onset dementia. I do see cerebral amyloid angiopathy. I had one person that was following pre-COVID, that 7 years, one person. And ever since COVID and the vaccines have come out, now I have 10. So there's definitely an increase in that. And I'd recommend getting any MRI you are going to get. You have to get some with either the T2 weighted stars, such as proton density, GRE, or SWI, depending on what brand name MRI you have, is going to be what protocol that you'll need. But always ask for that. And the other interesting thing is I've had 5 ALS patients show up. Now, granted, that's also an increase that I've had prior to COVID. And all 5 of them have actually tested positive for IgM Lyme. And so I think that's something that we need to really pay attention to and look into as another subset of activation. I'm seeing a lot of reactivated Epstein-Barr, and I always check for that. And I'm always checking for Lyme in these patients also. All right, so that is it. I'll answer any questions.
Wonderful. Thank you very much. I'll bring in Shankara and Philip, and I'll be looking out for questions from the audience as well. So I'll let them go ahead and ask you some questions.
Okay.
Shankar, would you like to go first?
Okay, Diane, I'm mostly dealing with acute onset COVID, and I've seen the sudden onset of psychiatric and neurologic symptoms. And my protocol for treatment tends to address a few issues, the cytokine release of histamine and leukotrienes, platelet activating factor. And so I deal with it as partly immunology with the antihistamines and that kind of thing. And of course, deal with microvascular clotting and try and prevent that. And of course, maybe a short course of steroid to just decrease inflammation. And I get some pretty remarkable results in a short space of time. But when you're dealing with long COVID, you're dealing with persistent neurologic issues that result in permanent injury. Now, do you find with treating long COVID patients that addressing that kind of inflammation and immunology tends to show benefit? Why I mention that, I've seen some strange results. I've had patients with sciatica that has resolved over the past 10 years. When they get COVID, the sciatica tends to resurface, but the traditional medication doesn't work. But when you address it as a cytokine release on a previous scar tissue and start to go down the road of anticoagulation and immunomodulation, you suddenly see benefit to it. I had the same with me with neuropathy in my spine, which I treated in a myriad of ways and nothing worked. But a simple antihistamine and Montelukast seemed to have done the trick. So, with the inflammation that you see in the brain, is it the cytokine release that needs to be addressed and is the triggering factor for the long-term damage if unaddressed?
Yes, I think it's one of part of what's going on in the brain. And so I really kind of hone in on a lot of their other symptoms to see what I really need to kind of heavily treat at the moment. I'm finding a lot of people will get supplement fatigue. I could come up with 30 different options that they could start right then and there, but I have to, pay attention to cost and again, pill fatigue. They just don't want to take a whole lot of things. So if they have a lot of mast cell activation type symptoms, I will definitely add in a lot related to, the cytokines related, famotidine, a type 1 antihistamine. Sometimes I'll even add in cromolyn. I'll also try to treat the microclotting, using nattokinase. And ivermectin is always my mainstay. The low-dose naltrexone I find to be very, very helpful. I think that's great. It helps with not only mast cell activation, but also the neuroinflammation in general. It will, work on many, many different receptors. And if someone has pain related to their neuropathy that they've developed, it helps with that too. And I've found a lot of treatment for dysautonomia. We are targeting mast cell activation to treat dysautonomia and getting beautiful results with that, with just some over-the-counter supplements, in addition to what we already mentioned. So I kind of give them a 4-product amount. I use a product called DHIST and GI Detox, which is a binder, plus the famotidine, plus a type 1 antihistamine, and then put them on a low histamine diet. I've been using a vagus nerve stimulator that you can get online now, over the counter, and to help increase their competency of the vagus nerve. And that seems to have been very, very helpful for the people with dysautonomia. So, yeah, but I'm targeting multiple areas, inflammation, mast cell, microclotting, and then just brain health in particular.
Are you ready, Bert?
Yes. Hey, yeah, Diane, on your experience have you followed many patients getting a pre and post NeuroQuant, and have you seen anybody improve volume wise?
Yeah, so one of the cases I showed, that she did have improvement in volume. I'm not ordering them very frequently because I think that would be, one, insurance would probably limit that, and then two, it's going to take a long time for the brain to heal. So this lady was 8 months out. And so I'm now kind of in the queue to start ordering more and more repeat MRIs and NeuroQuants. And I'm doing that on a more regular basis. And I want at least 6 months under my belt, to repeat the NeuroQuant on these patients.
Okay, shifting briefly to a different subject. Topic is about the brain, but I think you've been involved with or maybe played a part in, just to let the public know about glioblastoma and the antiviral that you would like that has been given, and we've seen some response with that. Can you briefly speak on that?
Oh, wow. Yeah. So there's a colleague that had a GBM, and it was small, did resection, and got most of it out. But of course, that it can, have these little extensions and legs going into the other brain. But he actually put himself on, again, cyclovir. And so far, his MRIs have been completely cleared up.
Wonderful. Dan, can I ask an audience question here? Just going to drop one in from the audience here. This is from Lever. Why is nicotine not being implemented on protocols from practitioners on this panel?
So I've used nicotine here and there, and I think in my mind it is probably more helpful in acute COVID cases. And I think once you hit long COVID, the receptors have already been used, by the spike protein. And now the problem is that the spike protein is now intracellularly. And so the nicotine receptors may not necessarily be in use at that point. So I think acute COVID, yes, it has its utilization. As Joachim Gerlach has mentioned to me several times, there's 26 receptors involved in spike protein, and the nicotine would be one. So I think there's other receptors that you shouldn't just ignore. But no, I've had some people get great results. I think it's also a good new in place of, say, caffeine, especially if you get it, not in, smoking. But it can be very helpful as a nootropic. But I've had several people that they tried it and it didn't do anything. But that's how all the supplements and all the treatments have been. It's not one stamp fits all. I have to keep on, testing the boundaries. I tell people it's like peeling back an onion. We're going to try to, help with these symptoms and peel those back and then see what's left, and then help with those symptoms, peel that, and then take out the supplements that might not be, helpful, the medications that might be helpful, and exchange it with something else. So, yeah, I'm always constantly changing and testing the waters and trying to help, the specific symptoms in front and center.
Excellent, excellent. And so wonderful. Can we close this section now? Thank you very much for the questions. What we're going to do now is hear a quick word from our sponsor again. After that, we're going to go in the roundtable discussion. I'll be looking out for specific good questions from the audience there. In reality, if you haven't seen it, there are lots of questions, comments happening in the background, but we'll try and see if we can address some of them in a short period of time. So let's just hear a quick word from the sponsor, and then we will get into our open forum.
And so when COVID started on day one, that is January 2020, I think around the 18th, we dropped everything. And ever since that day, we've been working 7 days a week, practically around the clock on finding solutions. Everybody on that slide you see there, to some degree, are working almost either on a weekly or daily basis, or even in direct collaborations, in research collaborations, in publications, in lab research. And so, each one there you can see has significantly contributed either to treatment or to development or understanding the disease and other discoveries. And so it's a great honor for me to be part of that and to be able to mediate between all these groups. I'm kind of like the coordinating a little bit between all these domain experts.
Wonderful. Thank you all very much. So yes, this was absolutely fascinating conversation. So I have so many thoughts and so many questions myself, but I'll try not to hog the round table. I'll be here to try and see if I can direct any questions as well from the audience. And just checking, I know, Manan, you were making a few comments, so I figure you wanted to say something at the beginning. And any specific thoughts that you wanted to say?
Can you hear me very clear now?
I want you to come closer to the microphone, please.
Okay, can you hear me very clear now?
Yes.
Okay, good. So I was also following the questions and like trying to answer a few of them right in the live sessions, and they are wondering that why other infections like EBV, CMV, and even herpes, is getting activated after COVID, and especially long COVID patients. So we published a paper, in which we showed that count is important. That if you count a lymphocyte and the count is coming normal, you think they are normal. But in many cases, we see a condition called as functional lymphocytopenia. That means you would have a lymphocyte which would not function. And this is what spike protein and the infection is doing to the lymphocyte. It is causing a type of immune deficiency. When your guards are down, then every other virus would become a lion, come over and attack you. So this is a viral reactivation that they are talking in the chat and comments, is due to a type of immune deficiency induced by the spike, be it coming from the virus or unregulated high production. Yeah.
Excellent. Thank you. I would like to throw something in. We're working on a paper in the background, and hopefully we'll be nearly finished it. And one of the points that we're going to make, and I'll put it to you all here now, is that I had a breakthrough thought with regards to the neurological manifestations, and especially brainstem involvement when they looked at long COVID and NeuroCovid. I actually believe that the primary location driver for the inflammation is the choroid plexus in the brain. So we are dealing with fourth ventricle choroiditis or third ventricle choroiditis. And once you have them releasing inflammatory cytokines directly in that vicinity, it will then activate microglial cells and cause neurological inflammation. And this is part of the reason why I think that we can't see anything, because we don't look for it. I don't know if there's any test. Maybe a PET scan would highlight inflammation in the choroid plexus, but just thoughts on that concept.
I've actually had a couple of MRIs where the choroid plexus looked very, very big, abnormally large compared to what I'm used to seeing. And I agree with that. that thought process.
And since these fluxes, have got endothelial lining and a core of blood vessels, just imagine endothelium anywhere is to anywhere, and the spike will find it. But then fluxes, itself tells you that they are a network of blood vessels. So more the blood vessels, more the endothelium, more the is to more the spike.
Excellent, thank you. Anybody want to take on Kevin and these clots that I think I've been following now for about 2 years, talking about it. One of the recent things I think you had made reference to, Kevin, was this question about tin. And it was because some of the original research had identified tin in some of the samples. So I was making reference to that. But in your research, you haven't seen any evidence that there is any tin or higher levels of phosphorus or sulfur in these clots. Would that be about right?
With respect to the tin, I haven't seen it. I am using a different method, which is electron dispersive X-ray spectroscopy with SEM. Those that have claimed to have found tin are using a different mass spec technique. But I think if there was significant amounts of tin, the EDX would find it. Phosphorus and sulfur. Yeah, you could say that there is a stronger signal there. Again, I would like to get more data, basically, before coming down on judgment in this instance. But the tin, in my mind, it doesn't seem necessary to form these abnormal clots. So what is it? And the genomics we've done has found ambiguous signals with respect to some of the markers with spike protein from vaccine, the SV40 and the origin markers, but we still have many samples to work through. It takes a long time to do these tests, and trying to do that on top of other stuff in the lab as well. My primary concern is just that they are being ignored when it's such a overwhelming signal with respect to this type of proteinopathy pathology.
Philip, I believe that tin could be more geographic, because depending on what your water pipes are made out of, tin's used as a catalyst in the PVC pipe. So in this country they may not have that. In this country they may have it. So you may see a higher level of tin because it's just in our bodies. I mean, it may or may not be a factor, but it still could be in the body. So
yes. Anybody want to take on this question here? Should people eat natto kinase daily as protection?
If Diane wants to go first, she's the frontline medic here. So.
Well, natto, comes from strict Japanese diet, and they eat a lot of it. And there is a certain area in Japan that has 0 Parkinson's, ALS, and Alzheimer's. And people have alluded to, could natto be part of it? We know that natto does help as far as anti-amyloid. Works as a mild blood thinner naturally. So, being that it's natural, derived from a food, if you wanted to take on a typical Japanese diet, I don't think that there would be a problem with that.
Go ahead.
But since being an enzyme, natto kinase could break and chop the spike protein into different fragments. The research that we are doing now in Heidelberg is actually testing the spike protein in the presence of natto kinase to see that what type of peptides it actually breaks the spike down into. So if the sequences that it breaks into, the wonder job it would do is that it could break the spike into amino acid, but then we would not worry about the amyloid sequences. But if it chops it down into small segments, then one of those segments actually is like amyloidogenic. Okay, I don't know whether enzyme of this category in a normal person it's fine, but having a spikopathy and having a NATO could be we should do clinical trials, okay, and before we come to a statement that yes, it is safe in COVID or long COVID patients.
I'd like to ask Philip, in terms of the radiology experience, and I was very fascinated by the results being shown with this NeuroQuant, and I'm beginning to wonder, and somebody did ask it, I can't find their comment, but they said, would it be recommended for people to have a baseline NeuroQuant study to be able to use as a comparator in the future if they have ongoing symptoms?
Was that question for me or Diane?
Yeah, for you from a radiology point of view.
Yeah, I mean, I don't think that'd be a problem at all. I think that'd be a great idea, but I don't know who's going to pay for it. And, certainly our government won't pay for it. So no, any baseline study would be beneficial. Yes. And that's my opinion.
Shankara, from the real world frontline experience at a very broad level, what would you say are the main neurological issues that are just running in the background that can easily get missed?
I think, Philip, repeated COVID Omicron variant infections is a huge problem. A lot of, like myself, initially with Omicron, I had quite severe symptoms. But now when I pick it up, it's just a slightly stuffy nose for a day or two. But then a week later, I start to get the body aches and pains, and I see the immunological dysregulation. And if people are not quick to that, that'll persist for months. And people are presenting with short-term memory loss, difficulty learning, slightly dyslexic kind of symptoms. These are mild symptoms that are going unnoticed, but will steadily progress. So with all my patients, Philip, I always mention to them that when you get the flu, it might not be the flu, it might be just a bioweapon attack. And you need to be very cognizant of recovery. And recovery should be 100%. 95% is unacceptable. And with Omicron, because of the mildness of the immune reaction, yet the long term, people tend to disregard it till it's too late. So we're getting patients coming to us with symptoms that have been persistent for 3 and 4 months. Previously, when we had the first 3 variants, patients came to us very quickly because the immunologic dysregulation was very sudden and aggressive. So I think the broader population just needs to be very vigilant of getting the flu, which in the past was just a transient, something that you never really thought of. Today, it could cost you your life.
Oh, important point. And Manan, I just wanted a thought popped into my head with regards to the overlap between neurology and psychiatric illness. When we think about the kind of patterns in terms of the damage in the brain that is occurring, should we then also anticipate not just the neurodegenerative diseases, but neuropsychiatric presentations increasing significantly?
The answer to your question is simple. When you say neurological and psychiatric, the psychiatric part is a baby. The mother is the neurology. Because those parts of our brain which actually responds to emotions and self-care and even feeling of anxiety and depression, their core is located in the hypothalamus, the limbic system. So this virus is not sparing any other region. Okay, if a person have got these regions involved, then psychiatric symptoms would dominate. If other regions like motor system or motor neurons are involved, the motor symptoms would be leading to the sign and symptom. Like Dr. Shankara mentioned, okay, that If it involves the nerves and its myelin sheath, then the demyelination and neuropathy in form of that would be the sign and symptoms dominating picture. So you cannot separate rigidly both of them. And eventually, if it's neuroinflammation, it's going to grip every other area of the brain, including that part, okay, which is responsible for our emotional behavior or our feelings. Yeah.
Yeah,
But the numbers are growing. The numbers are growing. We see many patients like who come who visited us for the fourth time. On the fourth time, the psychiatric part is actually overriding the neurological part. So the parents and the family complain that he's behaving abnormal, is very sensitive, is very alert, and like for the bugs, and would say something is crawling on the skin. While you check it, there is nothing. And also, anxiety and depressions are way above the highest point, okay, in COVID and long COVID. Yeah.
wow. And Kevin, I just wanted to ask you, because when you talk about prions and prion diseases, I don't think the general public can have any idea how serious that is. Now, from a personal experience, because prior to the pandemic, I've always heard about, say, mad cow disease, CJD. Never come across a patient, never interacted with anybody who had seen a patient. And for me to actually have a patient under my care in 2021 who deteriorated within 10 days, I knew at that time this was going to be very significant over the years. The problem is that from a prion point of view, once it gets going, there is no way to stop it.
Mm-hmm. Yeah. Well, there are sort of bleeding edge treatments now with respect to engineered antibodies to try to sort of limit the impact. I think the more important point is that I flashed a quick slide, which was that there is this interaction between fibrinogen and the scrapey form of prion protein, and how much that is ongoing and causal to the extreme fibrinogen amyloids that we're seeing. I think a lot of work needs to be done in that particular area, and if there is perhaps a therapeutic intervention, it's probably at that interaction, because that's what the body seems to be engaging in some protective activity with this fibrinogen prion protein interaction. Again, it's very new research, and the scale and implications just aren't being addressed, I'm afraid. It's quite unfortunate.
Yes, that's serious. And the other one, Dan, that you had touched on that I feel sorry for neurologists about is that if they don't have an understanding of long COVID and NeuroCovid, their diagnosis of functional neurological disorder is going to go through the roof. What would you say to neurologists now at this point? Because they must already be seeing it and be scratching their heads on it.
Yeah, and they try to fit it into what I say, a box of diagnoses that we had pre-COVID. But when you look at the Parkinson's that starts post-COVID, due to spike protein, it looks a little bit different. You can always kind of pick up just a little bit something different about it, whether it's a fast onset, or they have numbness, shortness of breath, along with all their Parkinson's symptoms. So it's really, really important to hone in on the review systems. And we aren't going to have anything nice and concrete like the rheumatologists have, diagnosing rheumatoid arthritis, such as, you have 10 of these 15 symptoms, you have rheumatoid arthritis. There's over 220 different symptoms associated with long COVID and long vax. And so it's going to be very, very difficult for someone to, again, use something very fast and easy as far as diagnosis. They are going to have to dive into the amount of COVID they had, the amount of shots they had, when did these symptoms start exactly. It was Lieutenant Colonel Teresa Long that said her data was showing in the DMED data that neurological symptoms happen about 190 days on the mean after the vaccine that were given to the military. And so that's 6 months down the road. And people don't put two and two together with that big time frame. And so I'm always constantly, and I see that a lot, 6 months. Of course, it can happen earlier, it can happen later. And I have a feeling the longer we get into this, we're going to see it later. But yeah, it is 6 months that people start having those symptoms after COVID and or the vaccine. So you have to get a great history. It just starts with the history. And that's how you can make the diagnosis. People just gloss over it. And I almost feel hard for neurologists now trying to get into the heart of this, because I've been through this, for the past 5 years. There's so much information and so much that you have to pay attention to symptom wise, that if I started right now, I'd feel like I was drinking from a fire hydrant. There's just so much information. But if you start little by little and understanding, the basic pathophysiology, and then keep on, learning and what treats what type of condition and potential pathophysiology that patient has, then I think they can do it. But the functional neurological disorders is very discouraging.
If I could just jump in, I do think this was something that was a problem even before COVID, with things like myalgic encephalitis, this type of post-viral symptom. And I get many, but it's often that I receive samples where the patient has been given functional neurological disorder as a diagnosis, and it's a combination of them not finding a concrete symptom, or they haven't looked for the right symptom, and that's the last sort of box that they have to tick to sort of give some diagnosis to the patient. And in this instance, particularly with COVID, if you can show the UV amyloid burden with respect to microclotting, and that's something concrete that the patient can then take to their primary care physician and say, look, there is actually a symptom or a correlate here. And the problem is that the much of the testing that's done, it's all very automated, and the fireflavin staining sort of requires generally older histological techniques. Someone's got to sort of sit there and look through a scope and try and get a measure of what's going on. And it's not a complex test, but it's just not available at any great level. And I know I'm doing it. Dr. Jordan Vaughan is doing it. There's a few other clinics. The flow cytometry has sort of come online recently as a diagnostic. But for those that have been, I would say, mislabeled with functional neurological disorder, I would say get that test first. Just see if there's a microclotting issue, because we know it's there. It's more intense with COVID relative to other post-viral symptoms, for example, with Epstein. But there is a easy step to take in this instance for those patients. And I can't guarantee that it'll get a signal every time, but I would encourage patients to, especially if they've been given that FND diagnosis.
Philip, the problem is that most of these physicians are not even, the ones that didn't believe in it to begin with, and then we're still dealing with that. The majority of the mainstream medical community, it's hard to convince them They didn't believe it the first go around. Now it's very difficult to convince them that we're even dealing with this problem. So we're going to, like you said, the functional neurology stuff is going to go out the roof. And unfortunately, that's where there's only a few people like Diane and Dr. Gaza and whoever treats on that side. There's very few of them, and that really truly understand it. And what I believe you were alluding to, we're going to have a bigger problem because all these people are going to be thrown into that grab bag, and it's going to be a sad situation because there's only a handful of people that are really still believing or that truly understand what's going on, and it's very frustrating.
Exactly,
So,
Dr. Philip. They are waiting for this to come in medical books, in
right?
15
Like
years.
you alluded to earlier, you exactly said
Yeah.
it. It's not in a medical book, so it's not real.
Yeah, exactly. And I don't know if they know it or not. All these medical books come from the published literature.
Right.
It doesn't come from the sky as a food. So they have to open their eyes, and I agree with Kevin and Diane that I just would like to quote a line from that very famous TV serial House MD. He used the word that symptoms and lab don't lie. So if there is a symptom and a sign and there is a lab finding which is saying positive more than 2 times or 3 times, don't close your eyes. Okay, go to a place. Okay, if they don't know, it's called as PubMed, and just type long COVID, and then sit and have your coffee. Okay, and get educated. So that's the thing. That's the lie.
Yes,
So,
and
making
when it,
excuses like I'm too busy in my clinic. Okay, when would I read the literature? So reading should never stop. Okay, you can just put it to an audio. Okay, ChatGPT could speak those papers for you.
yes,
That
Philip,
education is needed.
go ahead,
Philip,
Diane.
I have a comment. I think the research is slow because we're asking them to find evidence that implicates themselves. So I think we're going to have to do this ourselves. But I have a question for Diane. With the neurology, neuropathy that we see, have you noticed any correlation between persistent gut symptoms, which we've seen with COVID and with Carlo Brogna's work, and in the effectiveness of treatment, the time to resolution, and whether patients relapse more significantly if they have persistent gut issues, and are the toxins in the gut actually a driver for neuropathy? Amyloid, leaky gut, neuroinflammation, triggering histamine release, cytokine storm. There's a lot that goes on in the gut that can have a global effect on the body. And have you noticed a correlation in patients between the neuropathies and the difficulties with a persistent gut issue coexisting?
Yeah, so I'm always treating the gut at the same time that I'm treating everything else. And in fact, sometimes I have to start with the gut because if I start adding supplements and they can't tolerate it, then I really, really know that we have a long road to go as far as treating the gut first. And so, yeah, and I explained to the patients that, if we can fix the gut, we can actually decrease the inflammation that is coming from the outside of the world to the inside of the body. And then kind of, again, layering effect. I think the neuropathies are from multiple issues. Could be an endovascular problem, could be microclotting, could be inflammation, could be direct damage of the spike protein located in the dorsal root ganglion, and of course mast cell activation too. And so, if again, I kind of hone in on what other symptoms does the patient have in addition to the neuropathy, and I will try to target those specifically. I'd love to treat all of it, but I can't with the patients, just being too tired of a lot of supplements. Even the patients don't understand. They want to get better, but they don't want to understand that, hey, I have 10 products here that I want you to start. That's not what they want. One pill and done. Can I take a multivitamin? I get all that, that question all the time. So I am treating multiple things at multiple times, and including the reactivated viruses too. That's another big part. And I'm seeing reactivated Epstein-Barr cause a lot of movement disorders. I don't know if anybody else has seen that, but once I treat the reactivated Epstein-Barr, all this, and it's not Parkinson's, but it's twitching and movement and abnormal, head turns and movements that I'm seeing. It's really strange.
Quick question, Diane, there. When you say treating the Epstein-Barr, are you using an antiviral?
No, I actually will use ivermectin. Ivermectin has been found to help with Epstein-Barr, and I will use high-dose vitamin C, monolaurin, and a product called phytocidal, which has wormwood, artemisinin, licorice root, cat's claw.
Wow, it's remarkable to hear a neurologist who is willing to step outside of that box. So who wants to take on this question from the audience? I'd saved it. What's the risk for healthcare workers exposed to the spike and still being exposed to the spike? Yeah, Philip, go ahead.
Well, doctors, it should be Sean Caddy. Because he's been hurt the most, but we've all been affected. Even as a radiologist, something that I've been doing for 30 years, I will catch myself having to look up stuff that I've totally forgotten, and it should be routine. And, I do have brain fog and I do have symptoms. So, yeah. And, I know of 3 people on this panel that have been injured and they've never even had the vaccine. And so, yeah, it's a real deal. It's a real deal for all of us healthcare workers. And we're exposed to it daily. We're not backing away. We're not shying away. We're not running away. But, yeah, we're all affected in some way or another.
Yeah, and another issue here is just around blood products. I don't think it gets discussed enough. I've seen in the last couple of weeks a movement for safe blood, and I want to say I can't remember his name. It's on Jimmy Dore in the last week. And I think that's a very, very good idea at the moment until this amyloid issue is really addressed and there's some sort of systematic test put in place to make sure that the blood is clear of spike and misfolded proteins. But right now it isn't, and that's a serious concern.
I think that goes to any human product. We're going to have the next problem with organ donation. Same thing.
Yeah.
Yes, wonderful. Listen, we're going to be closing out now, and what I'll be asking each person to do is to give a final thought. And the reason why I did that is that this question came up, and it was, why are we not talking about things people can do effectively? They said in terms of to clean their blood, but this is really what a lot of people are thinking. It's one thing to know that stuff is going on, but what do we do? And so that's what I want you to think about as we try and close out. We'll go sequentially, and I'll ask, since we haven't had, who would want to go first? And then, okay, Philip, you'll go first. Then who would
you
be
guys
second?
cannot see me. I'm also raising hand. Okay, so
go ahead.
Yeah.
Go
okay.
first.
So it's Philip.
And no. I would follow Dr. Philip. Okay. I was just making a joke that you can't see me raising the hand because my camera.
I want to go first because I want to go first because it'd be the easiest, and then I wouldn't have to try to follow everybody else. Well, the problem is it's a novel process, and it takes a while. It takes time to figure out the disease process, which I think we're getting a handle on and a grasp on, and then B, what actually works to fight that process. And Kevin's in the lab working and figuring this stuff out, and now we got to figure out what's to break it down. Diane's seeing patients. Sean Caddy has been on the forefront. He was one of the first guys that actually taught me a lot about the process of the disease. So it's a novel situation, and it takes time to get to the bottom line and to figure out the actual treatment. And a lot of people are moving in a lot of different directions, and eventually we will get there. We will not stop.
Wonderful. And Mannan, what are your thoughts?
If you can highlight the question, okay, so I would just like see and
Well,
answer it because
it's not so much the question.
no,
Is what is your takeaway? What final words would you give to
final
the
word is
audience?
that line, okay, clean the blood. I mean, clean the blood is not the answer, I'll tell you. Okay, I'll give you two evidences for this. If it's a long COVID due to infection, you have to address the persistence of the virus, because if you keep on cleaning the blood, this covered virus will replicate and it will fill the blood again with the spike. Number two, if it's a vaccine issue, okay, and your cells are continuously producing it rather than stopping it on the 7th day, then you have to find a spike absorbent, something which clings to it, okay, and excretes it from the body, or target those cells which are overproducing it so that the spike doesn't spills in the blood. So clean the blood is an attempt. That attempt is not going to address the spike level. In order to address the spike level, antivirals for long COVID due to infection and some therapy which would stop the production of the spike by the cells which are overproducing it. Last week we patented something on this, the second one, how to halt the production, but infectious thing would need products like herbal medications until and unless some magic bullet lands on the ground. Thank you.
Wonderful. And Kevin, your final takeaway, what would you be saying to people now?
Well, I've been sort of predicting this cascade of neurological effects from the beginning, and it was something Mannan mentioned it earlier as well, that it was just dismissed out of hand as people thought that this was just a respiratory virus rather than being the systemic coagulopathy that it seems to present at. And the evidence continues to point to sort of mass brain and central nervous injury. With respect to techniques and cleaning the blood, it makes sense to me that you would do it. I recommend it to people if they have access to it, to try to limit the amount of amyloid. But as Mannan said, we don't know if that's a permanent solution or something that needs to be done on a sort of rotational basis. There's still much to find out in this instance. I would like to see more resources put into this, and a change of administration so far has not left me feeling hopeful. I'm fortunate to say, and I feel that the patients are being left behind as they.
Wonderful. Yeah.
We really want to talk about food coloring, or do we want to talk about the mass casualty events that has taken place?
Wow. Yes, very sobering, Kevin. And Dan, what are your thoughts, takeaway for the audience?
Yeah, it's not just the blood. Spike protein, once it gets into the body, be it infection or vaccine, it will float around in the blood and then find receptors and then go intracellularly. So you actually have to clean up the cells themselves. I've been using medicinals for that point. They sponsor that shows the liquid, but there's actually a 5-stack that I use, and there's some good information that shows that it could potentially extract that spike protein out of the cells, and then from there it goes in the blood, and then you use the binder to then excrete it out in the feces. So I think that's the big step that you have to do: get it out of the cells, and then you can extract it and then get into the problem. Second thing would be that COVID is going to act like the other viruses that have been reactivated. And so that's why we're going to continue to see this on and on again. And so we're going to have to come up with a prophylaxis. Me personally, I use ivermectin because I'm exposed to people that are shedding on me. When I go on, trips, planes, always, double dose on my ivermectin. Knock on wood, I haven't, gotten illness from that yet. So I still use a lot of ivermectin for that point, and don't forget that Yale study came out and showed that you are still producing spike protein from the vaccine as long as 709 days after the last vaccine was given, and still counting. Now, not everybody is doing that, but that was their data. So, we don't know where that goes to zero. So, we have that concern too. Are you still producing spike protein? So, I wish it were as simple as just clean the blood. People have used EBU, but that has tended to be a temporary measure for the moment, but then the symptoms usually come back. But I think that could be used part of it, but I don't think it's going to be the answer 100%.
Wonderful, thank you. And Shankara?
I think from a clinical perspective, Philip, I think there's two areas to address, the patient, the individual, and of course the medical fraternity that needs to be solving the problems. From the patient itself, I think there's a lot of complacency about circulating virus, and they think it means nothing. But I tend to educate my patients. The way the spike protein causes pathology is very unique, seems to have a phenotypic propensity. And when you react once, you tend to react the same way the next time. So I advise my patients to be cognizant of their peculiar reactions and bring it to the doctor's attention when they see it that second time. So I tend to educate them about how they specifically react to spike protein, and to be vigilant if an infection persists for longer than is usual, to be vigilant of the reemergence of those symptoms and a different modality to actually suppress it timeously. From the medical fraternity, with the mass vaccination campaign, we have negatively influenced the physiology of the entire planet very differentially by influencing immunology, which is very unique to different individuals. And so now the clinical presentation becomes very varied and, of course, confusing for that medical fraternity. The problem is that the medical fraternity is partly to blame for what has happened, and they don't seem to want to take the time to learn and figure out how to help. So there are a few of us that are carrying the can. So I think moving forward, it is the general population who has to now determine who the true medical fraternity actually is after what has transpired.
Wonderful. So thank you all very much. Much appreciated. We still have almost 630 people still with us on the live. So this is 2.5 hours. So they've really listened through. We really appreciate you all being here, asking questions, making comments. And we need this information to be shared. So remember to like, comment, subscribe, follow us along, and raise awareness. There is still a long road ahead. So thank you all very much. I'm going to do our outro, and I'll ask you all just to stay in studio for a few more minutes until after this is over. Thank you everyone for being with us, and look out for our next conference in the next few weeks to a month. Okay? Bye-bye.
Bye-bye. Bye-bye.
Thank you.